Okay, so welcome to our data contributors track portion of today. We're going to start off with a presentation by Laura Pearlman on data science practices and guidelines on writing data management sharing plans for FaceBase. I'm going to stop sharing so she can get started.
Okay, let me just cue this up. Just 1 second. Okay, so this is basically going to be about planning for data management and kind of why bother plan in advance. Partly it's because there are new requirements from NIH for people receiving NIH funding that they have a plan for how they're going to manage and share their data. But part of it is also to just make sure that your data is reusable, that you use data formats that other people can use, and that there's enough metadata that people can understand what your actual data is, enough to reuse it, and that metadata is expressed in ontologies that have standard meanings. So again, that it's understandable and reusable. And another reason to plan in advance is to just understand the level of effort that's going to be required and budget for it so that you're not blindsided when it's time to upload your data and you find out that you need to structure it in a certain way and add all this metadata that you didn't know that you needed. Okay, so some considerations are just when and how often you'll upload your data. We encourage people to upload early and often for a couple of reasons. One is that if there are any issues that come up with the upload, either technical issues with the upload or something about a metadata requirement that you didn't realize that you had, or some issue with a term that is not in our ontology, you can find that and catch it quickly when it's easier to fix, when it's immediately after you collected your data. For FaceBase, we do need you to have collected some data before we approve your upload, just so that we know exactly what kind of data that you have. Another question is how you'll manage your data before it's uploaded, how you organize it, making sure that you have sufficient space to store it before it's uploaded, and then looking at your security and backup policies, especially if you have controlled access data. And then there's a question of any pre-processing that you need to do, and what kind of tools will be involved in that. If you're developing themselves, are you going to make those tools public so that other people can use them? We have some specific considerations. We have specific data types that we support and file formats that we expect. We have metadata requirements, and we have preferred ontologies. We'll get into all of that. We are constantly evolving and expanding. So if you have data or metadata that don't quite fit this model, talk to us. But the farther away you are from what we already do, so there's a more lead time that we'll need to have those discussions and make those changes. So we have a list of currently supported data and experiment types and the species that we have in a document called Key Concepts for Data Contributors. And at the end of this presentation, there are links to all of the different documents and web pages that I reference. So this is not an exhaustive list, but just an example Some of the data types we have are sequencing, imaging, and dense surface data. And actually, you've seen all this in the earlier presentations today, so I'm actually not going to go through this. In terms of data formats, again, this is just some examples of the types of data formats that we have. The considerations that we have for formats are we want open formats, preferably, either because there's an open standard or de facto openness, and formats for which there are free and widely used tools available. And again, this is because our goal is to share data that is reusable by other people. We have specific metadata requirements. We want each experiment to have a protocol, and we recommend but don't strictly require the nature protocol exchange format. There's some other basic things: species, developmental stage, anatomy, and then each individual data type has additional requirements, again, to make that data usable. We support lots and lots of optional metadata elements, and we encourage you to provide as much metadata as possible because, again, it helps reproducibility. It also helps discoverability. If people are searching for things and you have the appropriate metadata terms, then it's easier for them to find. And we love metadata, so if you want to provide more than we currently collect, talk to us, and we will probably accommodate those additional elements too. And then we do use standard ontologies, and again, that helps with discoverability. We want to have, only one term for each specific concept. Here's just some examples of the ontologies that we use for FaceBase. I'm not going to go through these, but we have a lot of them, and each of them is in there for a specific reason. We do add new ones when necessary, but we do want to make sure that it's standardized and widely used. We have a process where we, talk with the community and make sure that it's a good fit and things aren't already covered by other ontologies that we already support. So that is the general overview of things that you need to plan for. I'm going to switch now into writing a data management and sharing plan, which is something that you will need to do if you're applying for NIH funding. I'm going to jump out of this presentation for a second because there are a couple of other things that I'm going to refer to. This is a document that is provided by NIH. Oh, no, it's not. This is a document that is provided by NIH that is a standard template for DMS plans. And I'm going to be referring to this a lot into the different sections. But basically, this is a Word document and it has the different questions that you need to answer. And so I will be referring to sections here. And then this is something that we provide on our website. This is a work in progress, but it has some text snippets and other information that you can use when writing your DMS plan for FaceBase. And now I'm going to jump back into my slideshow. Okay. So I'm just going to go through this kind of question by question. So with the DMS plans, they're looking for basically two kinds of things. One is the data and metadata that you are providing and information about that. And then the other is information about the repository where you will be keeping that data. So a lot of the questions in this DMS form cover kind of a little bit of both, what you're generating and where you're keeping it. And so in terms of writing the plan, there are some things where you'll have to provide some information, some where FaceBase will provide some information. And usually, in most cases, it's a combination. So starting out with a type and amount of data to be generated, that's all you. The amount of data to be shared and the rationale. again, in terms of the generating the data and what the data is, you'll provide that part of the answer. And then we will provide some boilerplate text about what happens when it's on FaceBase, how public data gets shared and how protected human data gets shared. In terms of metadata, depending on, so basically FaceBase has requirements for metadata that we've talked about earlier. So we provide boilerplate text snippets about the required metadata, the optional metadata, and then you can use those snippets to sort of put together an answer. There's a question about specialized tools and code and software that are needed to access and manipulate your data. If there are any specific tools that you provide or that you think people need, you'll need to provide that part of the answer. FaceBase's contribution to that is some boilerplate text about tools that we provide for specific types of data. So Rob showed earlier, for example, tools for showing annotated imaging data. And we have, browsers for sequencing data that are also mentioned there.
So Laura, just real quick, there's about 4 minutes left for this, including Q&A.
Including Q&A. Okay, I'll talk fast. Common data standards. We provide snippets for the standards that we support. You put them together to create answers for all of your relevant metadata types. And then some of these, we just provide the answer. We have our URL for FaceBase. There are questions about making the data findable and identifiable. And so we generally provide tools for that, capabilities for that. And then when and how long the data will be made available. Our policy is basically that we want the data to be available at least after it's published. You can also do it as early as after the curation process has completed. And then how long it will be available after it's published is something that we provide. We also provide some boilerplate text about how public and protected human data are shared and controlled. And then there's another question, I think at the end, about how compliance is going to be managed and monitored. And again, we will provide the text about how that happens once the data is on FaceBase, but you need to provide the text about how compliance is monitored at your institution. And okay, so further resources, this is a bunch of links, and I think that these slides will be made available on the FaceBase website, so you'll be able to find that. So NIH has a really nice amount of data about a nice site about writing these plans, including some templates and some sample plans. And then we have some FaceBase links. Every month we have office hours. So if you have questions, you can come and talk to us then. They're on Wednesday mornings. Chris has already put the help mailing address in the chat, and we love to talk to you about this stuff. And so please contact us at any point, and definitely once you're funded, because we want to know what data is coming in. Okay, so I can take questions now. Sorry.
Okay, great. So thank you, Laura. And if you come up with any questions afterwards, definitely feel free to email us. And as Laura mentioned, we will be sending out the webcast and the slides for all of today's presentations today, later today. Okay, great. So next up, we have our tutorial of contributing data. And Rob Schuler will kick us off with some overview information, and then Alejandro will follow through with a demonstration.
Okay, great. Thank you. Maybe someone needs to mute in the background, please. Okay, so I'm just going to give a quick overview on contributing to FaceBase and hand it over to Alejandro for a tutorial hands-on of the site. So first of all, you've probably seen already from the user session that FaceBase is a very diverse overall collection of data spanning human subjects to various animal models, from sequencing data, transcriptomics, to GWAS data, microscopy, micro-CT, and many other relevant experimental modalities that are useful for craniofacial and dental research broadly. And of course, there's a common theme among them, craniofacial, dental, developmental data, but within that, it's very broad. The benefit of sharing your data, aside from now it's being mandated, is that your data become first-class research products. You cite them like any other publication. They have their own scholarly life cycle, if you will. So the process for sharing data with FaceBase, and we really welcome broad contributions. The process begins with just letting us know a bit about your research and the research data that you've produced. So we have a form on the site. You'll find it under the facebase.org site under the contribute menu. And you go to this and give us a brief overview of your data. Tell us a bit about the type of data. Is it sequencing data? Is it imaging data? How much is there? What was the what were the objectives of the experiment, perhaps? Or even if it's just, we would be happy to collect, say, just data for the sake of having control data on a particular animal model that is not so common, or within the gaps that we have in our collection of data. So it could be really a variety of reasons that bring you to FaceBase to share your data. Once you've filled out this form, it is reviewed by the FaceBase Steering Committee. This is members both of the Hub, FaceBase Hub project itself, as well as our program staff and others who are members of that steering committee. Expect a response in about 2 weeks. We internally meet as a steering committee every 2 weeks. Every once in a while, there's a reason that the steering committee can't meet, and so a response could take longer. If you don't hear back from us within the 2-week period, just send an email to the help@facebase.org address, and we will get back to you with where we stand. So occasionally we may have additional questions about the data, especially if it's human subjects data. We may want to know more about what it was, how it was consented, and maybe some additional details along those lines. Once your data set is approved, and it usually is, then the process is what we call a self-serve data submission guided process. We like to begin with a hands-on tutorial with you. Now, coming to this meeting, you'll already have an idea of what that's like, but we like to tailor that to each site, each group that's contributing data. Then you use online forms, which you'll get a demonstration of. These are very spreadsheet-like in a way. They're organized around the attributes of each concept in our database, which I'll remind you again of what that looks like. But you get user-friendly forms to do that. They have bulk entry, a lot of things to help you enter from small one-off data collections to large ones with many biosamples and many data elements in them. We also have desktop tools for uploading files to the site. If you have, say, large collections of data, that can be helpful. We even have command line clients that operate off of, can operate off of your Linux workstation. If you're doing, say, transcriptomics with a large number of data or very large collections of data, usually you'll have those on a server. So if you're doing that type of research, and we have tools to help you do the import of that data as well. We have automated quality control rules that you can check back each day to see whether there are missing parts of what you've described, missing data elements, missing, say, biosample records, etc. And then when your data is getting close to being done, or where you consider it done, we, the hub, will take a look at it, and both members of our data team as well as our craniofacial subject matter experts will take a look at your data set and provide feedback. And sometimes these are things as simple as like providing a more informative description, or maybe noticing some keyword tags that could be added to your data set. And we like to help you through the process of finalizing your data set. Towards the end here, okay, thank you. Towards the end here, we suggest that while you're preparing a manuscript, you begin this process. And that's generally the case, that you'll be in the process of preparing a manuscript. So we generate the data citation information for you right up front as you create the data set. And please include that in your manuscript so that it is part of your released publication. And then finally, we can time the release of your data set with your publication. So oftentimes that's a very normal request, that one wants the data set to be released in sync with their publication. Okay, and then this is again, just to look at our share and cite feature that we looked at before, but I just want to mention again, as I said, plan this when your manuscript is in preparation. You can embargo it. Again, use the same citation format, even when you're citing your own data. And that makes your data availability statement more formal and easier to follow, and where somebody who is reading your article will know exactly where to go to get the data. And then again, looking at the detailed display of a data set, let me just point out a couple things of particular interest when you're contributing data. So highlighted in a orange there on the left-hand side, again, the attribution is listed clearly for each data set. This is for you, the contributor of a data set, not for a FaceBase team, but for your team, so everyone knows where this data set came from and who deserves the credit for any kind of citation to the data. And then occasionally, data sets that are contributed to FaceBase also have complementary data submitted elsewhere. So there could be genomic or transcriptomic data that's at Geo or at dbGaP, for example. Could be at other places as well. And we allow you to cross-reference those from FaceBase. And we would also encourage you to cross-reference your FaceBase data set from other sites. So sometimes we'll have a team or a contributor who has data that's on Geo or dbGaP, but they also have imaging data that they want out on FaceBase. We can accept both. It doesn't have to be split, but if you have a other obligation to share data on another site, you can share complementary data on FaceBase, so we support that. Okay, and just a reminder of what the structure of a data set looks like. Alejandro is going to take you through how you enter in data for this data model. We actually set up the project at the top for you, and then from there, you create a data set, which is the collection, and you describe your experiments, biosamples, and upload files and your protocols. As you saw before, again, we support a variety of visualization tools. We actually have the capability of generating a number of these visualizations automatically. Some of them have to be contributed by you, and we would really suggest consider, if you're contributing data, we do want the most raw or original data that was acquired off of your instruments, but we also really encourage the submission of some of your process data that can be more easily visualized on the site, because it really enhances your data set. All right, just a few quick tips here, and then I'm done. So we would distill down the basic strategy of a successful contribution is one, something that you just saw on Laura's slides, review the metadata and data guidelines early. So actually, when you're conducting experiments and collecting data, be aware of the standards and the type of information that you'd be submitting to your repository of choice. And we would hope that's FaceBase, but really any repository you're submitting to, you want to understand what you're going to need for the submission when it comes time to deposit your data. To take our 1-hour tutorial with us before you begin. That way it just really dispels any kind of confusion that you might have about how your data exactly fits into FaceBase, and we really tailor that for you. Third thing, you might even consider submitting, not submitting, but at least curating a part of a partial data set, like just enter one experiment and biosamples and data and have us take a look at it. We're really happy to provide feedback at any point in the process. And fourth, we're always here to help. Your main point of contact there at the bottom is typically going to be Alejandro Bugacov, who's going to speak next, but use our help at facebase.org because that goes to Alejandro, myself, Chris, and several more of us on the FaceBase team. So with that, thank you, and I'll turn it over to Alejandro.
Okay, thank you, Rob and Laura, for the introduction. I'll share my screen now. Very quickly, can you hear me and see my screen?
Yes.
Yes.
Okay, so I'll try to go through a quick tutorial about how you will rapidly build a data set. So like a little bit of Rob already mentioned this, but the process starts with submitting this data submission form that gives us information about what data you're submitting. Then the steering committee reviews that. Once that gets approved, it tells us, yes, we want that data. So we basically create a project for you, and we send you information on how to log into your project to start creating data sets. And also at that point, we tell you we would like to set up a Zoom call with you, typically a 1-hour introductory call where we'll go in a little bit more detail on what I'm going to do today. But not only we show them how you will create a data set and how you will upload the data, but the main reason why this call is important for us is because we discuss exactly what kind of data you're going to submit and how we think it will be better for you to organize this data, because like all these cases are very complex, very large number of different type of data and stuff like that. So there's always more than one way of doing these things, and it's interesting for us to discuss that so that we end up with a better possible data set. So I will show you the new projects by default are not public. Only you that has access to that data set and us can actually see that data. So everything you do in the beginning and everything you do until a data set is released, that typically takes a couple of interactions and iterations, then nothing is public. Only you have access to that. So that's completely protected. So once you get that, you just basically start creating data sets. I want to say that we are always available for help by email, or we have this at this location. We have very detailed step-by-step documentation of everything we wanted to say. The last thing I want to mention again is about human subject data. A lot of what I'm going to talk about applies also to human subject data, basically creating a data set, adding the description of the data set, some of the high-level metadata. But what it is different is how we handle the actual data. For human subject data, the data doesn't get uploaded to our site the way I'm going to upload files now, but that the actual data goes to a different, separate server, and that's handled completely offline. So we interact with you and tell you how to encrypt that data and how to transfer that data to our dedicated server. So with that in mind, I will try to now show you, let's say, let's say that we all went through all this, we have our call, and we ask you, how's your data set? So let's say this is a very simple data set where we have 2 mouse samples of a particular anatomy, which is the root of molar tooth. Let's say we have 1 sample that is a mutant, that the genotype is LHX6 --. Then we also have a control sample. And then on those samples, let's say you perform 2 types of experiment. One was a fluorescent microscopy experiment that generated some images, and we want to upload those images. And then you also perform some single cell RNA sequencing of the mutant sample, and we want to upload that data. So let's go and see how you will do that. So the first thing I will do typically when we go to our project is, if you're going to contribute data, you'll probably start you will go to your project and you will find your specific project, and then from that project you will add a data set to your project. So let's say I'm going to add this to my demo, FaceBase demo project, and you will see that if I look for it, I cannot find that project in here. And that's because this project is not public and I'm not logged into my system. So I will go and log in using a user that I know has been added to that project, the same way you're going to be added to your project once you submit your information. And now I see that this FaceBase demo project that I want to use is there. So when I go to there, I see some basic information about my project. You can use project member, you can add project member, funding information, publication, protocols. I'm not going to go through that right now. And then you have in the dataset section, you have the data sets that have been added to that project. And these are from previous demos and bootcamps. I'm going to add a new one here. So I will click Add Record, and this will take me to the basic form to add a new data set. And the main part of that initial data set will be to add a title. So the title, we wanted this to be like a title of a publication or a presentation or anything, something that is as descriptive as possible of what is it in your data set, what type of data do you have, what type of experiment you perform, what type of analysis you've done, anything, because that title is the information on that title is all going to be used as keywords and will help the discovery and also the understanding of what's in the data set. So in this case,
Alejandro,
yes.
Can you make the page a little bit bigger,
Sure.
please?
Yes, thanks for reminding me. Yes. Is that good?
Yeah, thanks.
Okay, so I will add a title. In this case, Bootcamp 2023 demo data set of root molar root of molar tooth, and then the description, something that is as descriptive as possible. Feels like the description and the study design that you see these icons in here. That means that it's rich text, it's marked down, and you can add pictures, you can add lists, number list, non-number list, links. Anyway, it's rich text. But anyway, in this data set, we perform imaging and single cell RNA sequencing of PN 14.5 mouse, something like that. And study design, typically, if there are data sets that have many different types of experiments, then you can add some. If not, then probably the study design part, it's covered in the protocol. So you can always ask the uploaded protocols explains how we perform and then some other basic information, whether it's human subject protected data, yes or no. If it were, you will add yes. And then if you require a DOI, you will always say yes, because then at this moment, and we'll talk a little bit more about that later, at this moment, then we basically tell the system to assign a DOI to this data set. And then there's a nightly process that will run and will assign this DOI to your data set. So even if you haven't released the data or anything, then you, at this point, you basically already have a DOI that you can enter in your manuscript if you're preparing manuscript, something like that, and that identifier will always identify this data. Anyway, so next step is to enter some high-level information about the data set. For example, contributors. The contributors typically get populated by all the members of the project. It gets automatically added there. But if you want to add some scientists that work specifically in this data set but are not part of your project, then you can add them here. You basically click link record, and let's say I'm one of them. You can search for my name and select here. Or if you don't find, if it's a new person, then you can create new and enter the information, and that will add a new user to the site. Same thing with publications. We typically, here we basically, is a field that we have for publications that already have a PubMed ID. So here you just enter the PubMed ID of the publication, and that's going to be automatically referenced there. Protocols, you will see that this section will be automatically populated once we add the corresponding protocols to the experiments. Another section that we have here is the one of related data sets. This section is for, in case part of this data, let's say, has already been submitted to dbGaP or Geo, or part of, or if this data set is related to other data sets inside FaceBase or within FaceBase, then you can add that linking or referencing of this data to the other one. So let's say the relationship, let's say it's a dbGaP data set, and then here, enter the URL, and that will create a link to that dbGaP data set. Then the next fields, human, is if protected data, human subject data or not, and the data use limitation. This applies mostly to human subject data sets, so I'm not going to go through that now. But this is something we typically discuss when we make this meeting and we discuss the specifics of your data. The next ones, like experiment type, species, phenotype, gene, stage, anatomy. These are all what we call the high-level tags that, in part, you will see that a lot of this information is asked again at each specific experiment and each specific biosample. But here, what you can do is you can add more than one. So at a biosample, you can only add one species, or you can only add one stage or one anatomy. But here you can add additional information, and we would like to have as much information as possible here, because all these fields are later used for indexing the data and for data discovery. So I will start, I'll show you how you can add that. So I will start, let's say here, I say I perform fluorescence microscopy experiment, and then I also perform a single cell. So I will add that in here. I will add that the species is mouse. But then I will stop. I don't need to add all these other fields, but you can enhance as many as you can. But if you don't, they will get automatically populated once you enter that information in the biosamples. And I will go through that now. And I want to show you something that we have here. We have on the top of our page this button that says hide empty sections, which is very useful because this toggles the information and shows you what you already enter and and gets rid of all that clutter for all the other sections that we have that may not be needed for your specific data set. But anyways, I will toggle this back and so once I'm done doing the sort of high level information, what I will do is I will sort of go into what we call the structure of the data set, basically entering the biosamples, experiments, relating the biosample to experiments, and adding the data. And the way I will do that is I will scroll down here to our biosample section and click Add Records and enter my first biosample. So the first field in the biosample is what we call the local identifier, which is basically this is a free text field that any identification that you want to use for your biosample. We know that a lot of labs that we work with in the past, they do follow very precise schemes of how they want to label their biosamples and experiments. So if your lab is one of those, then please use that local identifier in here. So this will be maybe the identifier that you use in your lab notebook, or whatever scheme your lab follows. Just use that information in here. If not, here I will say that this is some information that I know. Let's say 14.5. I will start by entering the mutant first. The species, and this may be several replicates or just this one. I will put just, let's say that's my first replicate. I will say that it's a mouse specimen. It's a, let's say, tissue specimen. One thing that I want to stress, and I think that both Laura and Rob went through it, but I want to mention again, is that as you can see, all these metadata fields, we don't make any of them basically coming from free text. They're all coming by selecting a term from a fixed vocabulary. And for some of the terms are just internal terms, but whenever it's possible, whenever it's available, we use information from well-known ontologies. So, for example, gene, we have all the NCBI terms. I don't know, here I will just pick a random one. Genotype is actually a different one. Genotype, we understand it's a very difficult thing to sort of standardize in an ontology. So this is all internal terms, but still, this is, you don't enter free text, you select this from a fixed vocabulary. If none of the terms that are here matches the term you need, then you contact us and we add those terms to the vocabulary so that you can select them. So let's say in this experiment, I will use LH6 that we know that has been used before. Again, we can add strain, mutation, stage. I will use PN 14.5. Select it. Other metadata fields that we have is anatomy. Let's say, so again, here we have all the terms from the Uberon ontology, which is very, very comprehensive, very large number of terms. I will look for root of molar tooth. We have other dimensions, which is origin, phenotype, type of treatment, sex, if it applies, the liter that it comes from. Let's say it comes from liter 1, and we can add the date where this sample was collected. So at this point, basically, I finished entering the information of my first biosample, but I want to show you a feature that I have here of how we can very rapidly create a very large number of biosamples. We actually have data sets that have hundreds or even thousands of biosamples. And even though it looks like a very time-consuming to enter the information, our user interface allows you to very quickly enter this information. So for example, if I want to add my second sample, I just put here clone, and this will create, a exact copy, and I'm only going to change the things that I know that are different. For example, the next one is the control, and the only thing that's going to change is the identifier, and I know that the genotype, instead of LX6, it will be a wild type. So I will select wild type. And then I will click save and navigate back to my data set. And if you see that automatically the two biosamples that I created are now listed in my data set. So the next step is to create experiments. And like we said, we perform fluorescent microscopy experiments and single RNA sequencing. So we'll create two experiments of fluorescent microscopy, one for the mutant and one for the control, because we want to make it so that under each experiment, we basically have all samples that are either exact biological replicates or just one sample. So I will start by adding one experiment. The experiment is the same. We select all these different terms. So I will start by picking up the experiment type. The first one, I will look for fluorescent microscopy. The local identifier, same idea. If your lab has a scheme of doing that, or whatever you use in your lab notebook, you enter that in there. If not, you just create something that is representative. So in this case, I will use fluor mutant PM14.5. That means that is a fluorescent microscopy for the mutant. At this point, none of these fields apply to microscopy experiments, so I can leave them blank. And then I will create two clones, one for the other microscopy and one for the single cell. So in my second clone, the only thing I will change is I will call it control here. And then in my third, I will change the experiment type to single cell. RNA sequencing. And then I will also change the name of the local identifier. I will call it single cell RNA, and it's going to be the mutant. So I will leave that like that, and it's the same stage. So I will click save. And now if I navigate back to my data set, I see the experiments now appearing. So now if I go hide sections, I see that, my data set now, I can clearly see what I have entered so far, all the high level information, the 3 experiments, and the 2 biosamples. So now the only thing that is left is to link the biosamples to the experiments and then add the actual data files. And the way we link biosamples to experiment at this moment is by what we call in our system a replicate. So you basically navigate to your experiment, and in the replicate section, you add a replicate, and in this replicate, you're going to say which biosample, you will select the biosample that you want to associate to that experiment. So here, you click biosample, and this filters down to all the biosamples only the biosamples in your data set. So in this case, it's a mutant, so I will select the mutant, click save. Then I will navigate back to the data set. I need to go this is the part you have to go basically experiment by experiment, but you can go fast. I will go to the other experiment, repeat the same process, add a replicate, select the other biosample, click save, and then navigate back to the data set, select my last experiment, and again, add a replicate, select the biosample. I said that we only did that for the mutant, so we'll select the mutant. And here I'm basically done with creating what we call the structure of the data set. The only thing that at this point is left is to add the actual data file. So, it only took us about 15 minutes to do this. So basically we have a data set with 3 experiments, 2 biosamples, and we have here it shows you all the information about that. So the way we actually add files to the data set, the way we upload the files is by adding these actual data files to the replicate element. So we go to the experiment that we want to add the data, we select the replicate that we want to add the data, and here we can see that we can add different type of data. We can add sequencing data, process data, this is basically the one, the data that has been processed using some sort of pipeline, typically for sequencing data, track data also for omics data, imaging data, mesh data, array data, all different type of data. Let's say, and then we have 2 basically main ways of uploading a file. One is directly through your browser. Let's say you want to add an imaging file. You go to the imaging data, you click add records, and here you can select some basic information about the file, like anatomy, device, equipment model. You can enter some description, but basically is here where it says file, you click select file, and you can pick a file directly from your computer. Let's say I will want to upload this image. I'll click open. I will select the file type. Here it was TIFF. The day was submitted. Click save, and the files gets uploaded to the replicate. And now if I navigate back to my data set, I can see that I have added 1 imaging data. This is a TIFF file, so TIFF files don't get automatically displayed, but like we'll see later, we have a pipeline that once you upload a TIFF file like this, our pipeline will process it and will generate a visualization for it and thumbnail, and allows for annotation of the image. I can do the same, for example, for a single cell. I can go here and add sequencing data through my browser if I want. But what I want to show now is that we also have a way of uploading data in bulk. Basically, if you have hundreds of or so of files, or 50 files, it becomes laborious to do it on the browser, and also if there are large files, sometimes you might get disconnected and you have to restart the process. And in order to deal with that, we actually have our own tool that is very good at dealing with that. That basically, once you structure your files in a given way, you can upload directly, automatically, all your files to the corresponding replicates. And this tool is very robust. It checks that your file has been uploaded successfully. If you get disconnected, you can always restart it from where you left off, and it will allow you to upload hundreds of files. So let me show you how you will use that tool. So I will give you, first I want to show you a little bit. Here we have some documentation that shows you how you have to, how you will basically structure your data so that you can use this upload tool. So here in our documentation, we have this section that says organize your files. And this basically, I don't know if you can, if it's big enough, let me make it a little bit bigger. Basically shows you the organization of how you organize your files in your computer, so that once you've created this structure of the data set in our site, you can organize the data in your computer and then use our tool that we call the Deriva upload tool to upload all this data at once. And the structure that we use is sort of simple. So we create a folder that is the data set, and here is we enter what we call the record ID of the data set. I will show you now how to get that. Then under that folder, we create one folder for each replicate, okay? And then under that folder, we create any subfolder for the different type of data. So let's say if it's sequencing data or process data or imaging data, we create subfolders and we place the data in there. So let me show you now how I basically organize that. So let me go back to my data set. And so I will show you now how I can add some data to this data set. So the record ID of this data set is this. This is what we call the read. So I will copy that. And then I will go to my machine. I already have kind of prepared the structure. And then I will create this folder. I will enter.
Can you make that a little bigger?
Let me try. I'm not sure I know how to do that.
That's okay.
Okay, but anyways, I will create this folder. I will enter the name of this folder is going to be the record ID of the data set. And then I will create one folder under that one for each of the replicates. So I will go here now to the experiment, and I know the replicate ID for the fluorescent microscopy mutant is this one. So we'll copy that one, and I will go to here, and I will go imaging mutant. I will enter that ID. I will do the same for the control. Let me finish this and then I see if I can figure out how to make it larger if you guys cannot see it. And then the last one here is the replicate for the single cell data. So basically I have this structure where I have the folder with a read of the data set, and then under that 3 folders, one for each replicate, and under the replicate I have the different type of data. I have some imaging files, some thumbnails for that file, and then for the single cell data I have some process data where I have my barcode files, my features files, my matrix files, and then some also some of the raw FASTQ files. So once I have that structure, I can open the tool that we can show you how to download. It basically downloads and installs in all platforms. And it's very well documented. Anyways, once the tool starts, it already points directly to our FaceBase browser. The next thing I need to do is log in. I will log in with the same user that I logged in before. There. Which I believe was called hope I can authenticate successfully. Okay, seems like I'm logged in. So the next thing is I need to click here, browse, navigate to the top, to the directory with the top level directory that has the record ID of the data set. Click open. That scans all that folder and subfolders for all the type of data, makes some consistency checks, and then once you went through it, you click upload, and this should hopefully upload all the files. Okay, and that's done. And now if I go back to my data set, I hide all my sections. And now I see that I have uploaded some images with some thumbnails, and now all the the sequencing data that I have loaded, the process data for the single cell data, and the imaging data that I uploaded. So at that point, the only thing that is left is that we require all experiments to have a valid protocol. So basically, you will go back to the experiment, and here in the protocol section, just assign a protocol. So for example, let me start with a, if I go to the single cell experiment, I can add, click link and add, maybe I already know that the 10x genomic single cell protocol is there, so I can probably reuse that one. I can check if it works for me, I can reuse it. If not, you will create new, give a title, my 10x protocol, and then select the protocol type, let's say data processing, and here you can enter a description here, or you can give a URL if it's already been published, or you can, if you already have that protocol in a PDF document or something like that, you can select file and upload it from your computer. We'll click save. Well, for some reason I need to also select the, I will leave that now. But anyways, I just want to know that you can just select the protocol, and that basically at that point you're basically done. So we have, once you added all a protocol for each different type of experiment and uploaded your data, you're basically done. And what happens after this is that you let us know that you are basically done curating your data set, and we review it and we go through a Q&A process. And once you're done, we either release your data if you're ready for releasing it, or we can embargo your data and leave it ready when you feel that it's ready, that you're ready to make your data public. You let us know and we release the data to the public. Okay, so that's basically the creation of a data set. I don't know if you have any questions. No questions? Okay. Anyways, once we have that initial call with you, we go over all this in much bigger detail, better detail. So the last thing, if I still have time, the last thing I wanted to touch on is some of give sort of like a overview of the different type of data processing and pipelines that we have available. A little bit of that we already talked about. But one is the image, what we call the imaging pipeline, that basically once you upload one of these files, like I have shown already, let's say if it's it can be a single PNG or JPEG or TIFF or OME TIFF, or something as complex as a multi-channel 3D or 4D stacks. We have our pipeline that will run through it nightly and will basically create thumbnails and create a visualization that will allow annotations of those images. So you can, the user can manually go and tag different sections of your images with a different anatomical term, or enter text, or use arrows and mark regions. So that's one pipeline. The next one is the genome browser that we already, I think Rob mentioned a little bit, but basically if one of these files that I uploaded would have been a big WIG or a big PET file, then those files, I can directly add a visualization to the UC Santa Cruz genome browser, and that automatically shows in your data set. So you don't have to go through any of these sort of sophisticated configuration things so you can see your tracks in the genome browser. This, once you upload into FaceBase, you will automatically have this genome browser visualization. Similar is the cell browser pipeline. Like for example, I just shown, I uploaded the matrix file, the feature files, and the barcodes. We have a pipeline that runs right after you upload the files, and will create a cell browser visualization of those files. So right now it takes certain files and some output of the Cell Ranger pipeline, and we are always constantly trying to improve that. But basically, that's what we're targeting. The other one is we have a mesh viewer, like Rob already shown. If you upload a file in OBJ format, then we can create a model. You can directly have this 3D visualization. Same thing for our volumetric viewer. You upload a file in Nifty, we can, if the file is too large, sometimes we typically do a manual resolution reduction, then we can upload it, and then you have a automatic 3D visualization. And the last thing I want to touch is the assignment of DOIs. Basically, like I have shown already, once you create your data set, then at that moment you have a data object identifier for that piece of data. You don't have to wait until it's released or anything like that. That DOI you can directly include in your manuscript right away. If for some reason you need anonymous access to your data by journal reviewers, you can contact us, and we have mechanisms to do that. Our DOIs are assigned automatically for each data set and also for each project. So you can also reference your project with many data sets. We also generate links for every page that we have. At the top corner, you can go, I will show you now, click the share and cite, and that will provide one unique identifier and persistent that will never change for every page in our site. Same thing for our data sets and projects. Every data set and project has a unique persistent identifier that will never change. And in there, we also generate this, automatically generate this text that you can use for citation. And this is basically the way we would like you to cite your data in any publication or presentation with the correct DOI and the title and the source. So actually, let me show you that in here. If we go to this data set, here, we click share and cite, and that's where you will get that. So right now, you see, it says in preparation because it hasn't been, it's not released yet, and the DOI hasn't run yet. It typically runs a few minutes after that or overnight, and you will have also the DOI here and say in preparation. But all these links, that link, for example, is always persistent. You can always come back to your data set by using this identifier. And this is what we call a version. You know, as different versions change, we also generate different versions. Okay, I think that's, with that, I think I will probably finish my presentation. I don't know if you guys have any questions about anything specific.
So yeah, we're open to any questions from anything today that you might have. If you have data that you plan on contributing and there's anything specific to your data that we haven't addressed, or if you have any questions about any other procedures that we haven't covered.
So we'll be uploading some human data. So how different is the process? I know it like goes through a whole different channel.
Yeah, that's a great question. Yeah, so it differs at the point of basically uploading the data. You know, you basically go to our site, you create a data set, you enter the description, you may have to add the protocols, you may enter some information on experiments and maybe some samples or subjects. But then the data itself, everything that is individual level data cannot be uploaded. We contact you, we provide you an encryption key that you will use to encrypt the files that you will submit, and then we'll give you a way of transmitting those encrypted files to our dedicated server. And then that data lives in that server. So it's basically it's like a rsync or you know, FTP type of mechanism where you basically transfer the data from your computer or your server to another server.
Okay.
So you basically package it, encrypt it, and transfer it.
All right, thank you.
Yeah, but we would like you to create a data set and enter as much information as possible so that that data is discoverable, you know, so that people know that that data is there, and enter as much information as possible, like how many subjects, what type of data we have, all that information, so that someone that wants to request the data knows what's in there. And then typically after you submit your data, what we do is we create a manifest file that we upload to our site so that people know what are the files that are included in your data set and the size and the checksums, so that when people get the files, they can verify that what they got is correct and is the exact data that you uploaded.
Okay.
And one more thing, you know, any anything from your human subject data that is at the aggregate level or that can be shown that doesn't have any identifiers, then yes, you can discuss that with us and we can make that public.
Okay, yeah, I think most of it, I mean, we're gonna have a joint called VCF, but that's still gonna have genotypes in it, so that is still probably needs to be controlled.
So that basically is something that we discuss with NIDCR and us, and what anything that they tell us that it's okay to show, we put it on the website, and anything that they tell us that should not be shown, then we.
Yeah, I suppose we could just have a list of variants that we called in the data set, and if we don't have specific, like individual specific genotypes, that might be okay. That might help someone decide if they wanted it or not. Anyway, I'm just brainstorming.
Yeah, we'll typically discuss on each in each case.
So I'm muted. Okay, great. And then Navdeep, Rocio, do you have any questions or any, or if you just want to kind of let us know if you have some data that you're expecting to, that you would like to do with FaceBase, we have some time we could talk. Okay, but yeah, that was a great presentation, both Rob and, well, Laura, Rob, and Alejandro, thank you so much. And yeah, thank you all so much for joining us today. I'll just share this screen again. I shared this right after the users track. This is just us saying thank you, and that we're very happy that you joined us today, and that if you have any questions or comments, any other communications you want to make, please feel free to use our email, again, the email address help@facebase.org. And it goes to many of us from the hub that are on this call. So there's always somebody available who can respond to your question. And again, if there's feedback, if there's something in our documentation or in our process that you think could be more friendly or make things easier for you, we definitely want to hear about that too. So thank you very much for joining us. Let's see, we have. Yeah, thank you so much, Navdeep. And as I mentioned before, I'll be sending out the webcast and slides for today's boot camp and for your home viewing pleasure. And other than that, I think that'll do it for today. So thank you all again, and I'll go ahead and stop recording.
All right, thanks.